So good afternoon to everybody. Today we discuss about the glycine metabolism. What is the glycine? Glycine is an amino acid. Is it a non-essential or essential? It is a non-essential amino acid and simplest amino acid, but it is optically inactive and it is a glycogenic amino acid. So we can discuss later how it is a glycogenic amino acid. Okay, it is involved in the synthesis of the proteins, synthesis of the heme, synthesis of the purine, synthesis of the creatine, and also participate in the one-carbon metabolism. If you consume the excessive, that is glycine, so leads to the renal stones. When you consume the excessive glycine, leads to the renal stones. So it is a non-essential amino acid. Okay, so and it is a glycogenic amino acid. Says properties proteins. So glycine, where it is present in proteins. That is, glycine is present in the proteins interior structure. So this is glycine is stored in present in interior structure of protein. Example of the protein here it is a collagen. So collagen containing the very high amount of glycine, that is about 30%. So other than amino acids, collagen containing so very high content of glycine, that is about 30%. So if you see the collagen, every third amino acid in collagen is glycine. If you observe the structure of the collagen, every third amino acid in collagen is glycine. So glycine is involved in the protein structure. It is present in the interior structure of the protein. So because of it is very small and non-polar in nature. So about 30% of the glycine is present in the collagen. Now see the synthesis of the glycine. Glycine is synthesized from the serine, threonine, carbon dioxide, and ammonia and glyoxylate. So that means is nothing but is the sources of the glycine, serine, threonine, carbon dioxide, and ammonia and glyoxylate. Or otherwise is so requirements of the glycine synthesis, that is serine, threonine, carbon dioxide, and ammonia and glyoxylate. These are the things are required for the synthesis of the glycine. Or otherwise is glycine is formed from the serine, threonine, carbon dioxide, and ammonia and glyoxylate. Okay. Now see the synthesis of the glycine from various components. Okay. Glycine is synthesized from the serine. Glycine is synthesized from the serine. See here it is glycine. So serine involved in the formation of the glycine. See here it is the arrow mark. Glycine is involved in the synthesis of the or formation of the glycine by the here it is the reaction is catalyzed by the serine hydroxymethyl transferase. Serine is converted to the glycine or otherwise glycine is formed from the serine. This reaction is catalyzed by the serine hydroxymethyl transferase. Okay. So here it is the carbon glycine synthase. So this glycine synthase involved in to convert the carbon dioxide, ammonia and one carbon unit, that is N5N10 methyl tetrahydrofolate is a one carbon unit. So convert to the glycine. So glycine synthase involved to convert the carbon dioxide, ammonia and one carbon or otherwise is N5N10 methylene tetrahydrofolate is nothing but is a one carbon unit. So okay, so you convert to the glycine. Convert to the glycine. So the glycine is formed from the carbon dioxide, ammonia and one carbon unit. That is a one carbon from the one carbon full, that is N5N10 methylene tetrahydrofolate. Okay. Glycine also synthesized from the or otherwise synthesized from the threonine. See here it is. Threonine is involved in the formation of the glycine or synthesized the glycine. Here it is that this reaction is catalyzed by the threonine aldolase. This reaction is catalyzed by the threonine aldolase. Okay, glycine also formed from the glyoxylate. See here it is the aroma glyoxylase by transamination reaction. By transamination reaction. So the glycine is synthesized from the one carbon metabolism, and here it is a carbon dioxide and ammonia, and from the threonine and from the serine and from the glyoxylate. So the glycine is synthesized from the all sources of the glycine, glyoxylate, serine, threonine, and one carbon unit, carbon dioxide and ammonia. These are the sources for the synthesis of the glycine. Okay. So this glycine undergoes the degradation. What happens? What are the products of it? Degradation of the glycine. Glycine can be degraded to carbon dioxide, ammonia, serine and glyoxylate. See here it is. Glycine is degraded to carbon dioxide and ammonia. See here it is. and here it is again serine, and see here it is a glyoxylate. See glyoxylate. Glycine undergoes the degradation, produce the carbon dioxide and ammonia. So when undergoes the degradation, produce the serine, and here it is a glycine undergoes the degradation, produce the glyoxylate. See, here is some reactions are reversible reactions. See, observe the metabolism here. Some reactions are reversible reactions. The formation of the glycine from the carbon dioxide and ammonia, this is the reversible. That means this glycine again degrade to produce the carbon dioxide and ammonia. And glycine is synthesized from the serine. See, here it is. Serine also is involved in the glycine is synthesized from the serine. And serine is formed from the glycine. See, reversible reaction, here it is. So that means the serine is converted to the glycine, glycine is converted to the serine. Okay. So here it is a reversible reaction, check it here. So glycine is converted to the glyoxylate, this glyoxylate is converted to the glycine. See here it is. Glycine is converted to the glyoxylate, and this glyoxylate is converted to the glycine by transformation reactions. Okay. Now see how it is a glycine is a glycogenic amino acid. See, glycine undergoes the degradation reaction, produces this serine. This reaction is catalyzed by the serine hydroxymethane transferase. The serine hydroxymethane transferase is a tetrahydrofolate dependent enzyme. Tetrahydrofolate dependent enzyme. Okay. The serine is degraded to pyruvate. This reaction is catalyzed by the serine dehydratase. Serine is degraded to pyruvate. This reaction is catalyzed by the serine dehydratase. Okay. This pyruvate is a precursor for the glucose. This pyruvate is a precursor for the glucose. That's why glycine is a glycogenic amino acids, because of the, so it is involved in the synthesis of the glucose through the serine. Okay, so that's why it is a glycogenic amino acid. Now see the, here it is the glycine undergoes the oxidative deamination reactions. Oxidative deamination by the glycine synthase. See here it is the glycine undergoes the oxidative deamination reaction by the glycine synthase, produce the carbon dioxide, ammonia, and here it is a one carbon fragment. One carbon fragment. Here it is a one carbon fragment is example N5 N10 methylene tetrahydrofolate. Is a one carbon fragment. Okay. This glycine synthase is a multi-enzyme complex. For its activation, NAD is required, PLP is required, and here it is a tetrahydrofolate is required. So glycine synthase is a multi-enzyme complex. So when the examiner asks you what example, give the example for the multi-enzyme complex, then you have to give the answer is glycine synthase. So glycine synthase is a multi-enzyme complex enzyme. For its activation, it's required the NAD, PLP, and tetrahydrofolate. Okay, these are the requirements for the glycine synthase activation. Okay, now see the glycine is degraded to the glyoxylate. Okay, see the formation of the glyoxylate from the glycine, from also is a serine. Glyoxylate is formed from the glycine and also is the serine. See, check it here it is. So glycine is converted to undergoes the degradation, produce the serine. Serine undergoes the degradation, produce the after several reactions. Here it is glyoxylate. Okay, glyoxylate. Serine degrade to produce the pyruvate and also is undergoes the several reactions, produce the glyoxylate. See here it is the glycine undergoes the degradation, produce the glyoxylate. So the formation of the glyoxylate from the glycine and also serine. Don't forget. So formation of the glyoxylate from the serine and here it is a glycine also. Okay, so glycine undergoes the degradation, produce the glyoxylate and also is a carbon dioxide and ammonia, and here it is the serine. Serine. Now the serine undergoes the degradation, produce the pyruvate. This pyruvate is a precursor for the synthesis of the glucose, and also so serine undergoes the decarboxylation reaction. Here it is produce the glyoxylate after several reactions. Okay, this glyoxylate is undergoes a transamination reaction, reversibly convert to the glycine. See here it is, back to produce the glycine. This glyoxylate undergoes the transamination reaction and here it is the back to produce the glycine. This transamination is a PLP dependent enzyme. PLP dependent enzyme. Okay. So glycine degrade to produce the serine. Serine undergoes the degradation, produces here it is a pyruvate and also some glyoxylate. Okay, this pyruvate is entered into the is a precursor for the glucose. Okay, this glyoxylate undergoes the oxidation reaction, produce the oxalate and also is a formate. So glyoxylate undergoes the oxidation reaction, produce the oxalate and formate. This oxalate is an excretory product. Oxalate is an excretory product. But what is the form here? It is formate is combined with the tetrahydrofolate and enter into the one carbon fragment for the formation of the one carbon pool. Enter into the one carbon pool for the formation of the one carbon unit. So that is an N10 format tetrahydrofolate. So enter into the one carbon metabolism for the production of the one carbon units. Okay, that is N10 format tetrahydrofolate. This entire metabolism considers see here it is both the anabolism and catabolism. So both the anabolism synthesis and degradation. So glycine synthesis occurs from the here it is the serine, threonine, glyoxylate and carbon dioxide ammonia and from the one carbon metabolism fragment. Okay. Glycine is degraded to produce the again serine, glyoxylate and here it is a carbon dioxide ammonia and here it is the pyruvate. So this glyoxylate is again degraded to produce the oxalate. This oxalate is excretory product and glyoxylate again will degrade to the formate. This formate will enter into the one carbon metabolism. Okay. See the synthesis of the biological important compounds. Some compounds are synthesized from the glycine. These are the biologically important. See what are they. So glycine is involved in the formation of the purine ring, involved in the synthesis of heme, involved in the synthesis of glutathione. So and also act as a involved in the conjugation reactions and involved in the synthesis of the creatine. So here it is a glycine is most important for the formation of the biological important products. Here it is a synthesis of the heme, synthesis of the glutathione, conjugation reactions, synthesis of the creatine, and involved in the formation of the urine ring. See, the first one is involved in the synthesis of the urine. The entire molecule of glycine, see here it is the entire molecule of glycine involved in the formation of the purine ring. This is a purine ring. See here it is the N10 format tetrahydrofolate. So involved in the formation of the C2, aspartate involved in the formation of the N1, that is a nitrogen compound. So and here it is the carbon dioxide involved in the carbon 6. And here it is the N5, N10 format tetrahydrofolate. Here it is involved in the formation of the carbon 8 and glutamate involved in the formation of nitrogen 3 and nitrogen 9. So N3 and N9. But here it is the entire molecule of glycine involved in the formation of the carbon 4, carbon 5 and N7. So this thing is synthesis of the purine ring. So glycine is the entire molecule of the glycine involved in the formation of the purine ring. Now see then, another most important biological important one, synthesis of the heme. So generally, glycine is involved in the synthesis of the heme. The primary product of synthesis of the heme is glycine. See here it is a succinyl-CoA when combined with the glycine. Succinyl-CoA when combined with the glycine produce the delta amino levulinate. That means ultimately these delta amino levulinate undergoes the several reactions finally produce the heme. So that the final that is the initial product of the synthesis of the heme is glycine. So succinyl-CoA when combined with the glycine produce the heme. So these reactions occurs in the synthesis of heme occurs in some reactions occurs in mitochondria, some reactions occurs in cytosol. But ultimately, the final production of heme is occurs in the synthesis occurs in the mitochondria. See here it is. Succinyl-CoA when combined with the glycine produce the delta amino levulinate. Here it is a amino levulinic acid. This reaction is occurs in the mitochondria. Okay. So the initial product, what it may be, glycine is when combined with the succinyl-CoA. So this reaction is produce the amino levulinic acid. This reaction occurs in the mitochondria. Okay. This amino levulinic acid enters into the cytosol. It is converted to phosphobionesin. and the again is converted to urophosphorylation 3. This urophosphorylation is converted to coprophosphorylation 3. This coprophosphorylation 3 will enter into the mitochondria. Here it is converted to the protophosphorylation 9. This protophosphorylation will convert to the protophosphorylation 9. So this protophosphorylation 9 so convert to the heme. This reaction is catalyzed by the ferrochelatase. So ultimately the initial product, that is a glycine. So synthesis of the glycine with the succinyl-CoA. This reaction is occurs in its mitochondria. The final product is heme is also synthesis of the reaction is occurs in the mitochondria. So okay. So glycine is involved in the synthesis of the heme and also involved in the formation of the urine tree. Next one is glycine involved in the synthesis of the glutathione. So glutathione is a tripeptide. Don't forget, glutathione is a tripeptide. So why it is tripeptide? Because of it requires the 3 amino acids for the synthesis of the glutathione, for the formation of the glutathione. What are the 3 amino acids is required for the glutathione? That is glutamate, cysteine and glycine. Glutamate, cysteine and glycine. These are the required for the formation of the glutathione or synthesis of the glutathione. Okay, that's why it is also known as gamma glutamyl cysteine glycine. Glutathione also called glutamyl cysteine glycine. See here it is. So glutamate is combined with the cysteine, produces the gamma glutamyl cysteine. Gamma glutamyl cysteine. Here it is. This reaction is catalyzed by the gamma glutamyl cysteine synthase. That means glutamate when combined with the cysteine produces the gamma glutamyl cysteine by the synthase reaction, that is a gamma glutamyl cysteine synthase. This product, that is a gamma glutamyl cysteine when combined with the glycine. See here it is condensed with the glycine, C attached to the glycine. So here it is the gamma glutamyl cysteine. Now the glycine is attached with the gamma glutamyl cysteine, produces the glutathione. That is nothing but as a glutamyl cysteine glycine. Glutamyl cysteine glycine. This reaction is catalyzed by the glutathione synthase. This reaction is catalyzed by the glutathione synthase. So here it is a glutathione is a tripeptide. It is required for the formation of the glutathione 3 amino acids. That is 2 G's and 1 G. See here it is a C. So 2 G's means glutamate, glycine, and C means cysteine. These 3 are required for the formation of the or synthesis of the glutathione. See the functions of the glutathione, important or otherwise importance of the glutathione. Where it is distributed? It is widely distributed in tissues and it is present in 2 states, that is oxidate state and reduced state. It is present in oxidate state and reduced state. It is a main component of an antioxidant enzyme. It is a component of an antioxidant enzyme. Example of the antioxidant enzyme, here it is glutathione peroxidase. It is a component of the glutathione peroxidase. So what is meant by the antioxidant enzyme? The main function of the antioxidant enzyme is scavenges the free radicals. It scavenges the free radicals and also maintains the integrity of the RBC memory and protect the hemoglobin. Here it is hemoglobin from the oxidation by hydrogen peroxide. It protects the hemoglobin from oxidation. So if this oxidation occurs by the hydrogen peroxide, that's why it protects the hemoglobin from the oxidation by hydrogen peroxide. And also involved in the transport of the amino acids, that is a Meister cycle, involved in the detoxification reactions, and involved in the formation and stabilization of the disulphide linkages in proteins. So glutathione involved in the formation and stabilization of disulphide linkages in proteins, and also act as a cofactor for some enzymes. The main here is the widely distributed in all the tissues. It is existing in 2 states, there is the oxidate state and reduced state, and act as a component is a antioxidant enzyme. The antioxidant enzyme example is glutathione peroxide. The function of this glutathione peroxidase is scavenges the free radicals and also glutathione involved in the maintaining the integrity of the RBC cell membrane and also protect the hemoglobin from the oxidation by the hydrogen peroxide, involved in the detoxification reactions and stabilize the formation and stabilize the disulphide linkages in proteins. Now see the conjugated agent. Glutathione is a conjugated agent. Sorry. Glycine is a conjugated agent. So then immediately you have to remember that is a formation of the, or otherwise a synthesis of the, here it is a folic acid and deoxyfolic acid, that is nothing but is bile acids. So bile acids are formed when conjugated with the glycine. So bile acids are formed from the conjugated with the glycine, then it is formed. Okay, bile acids. Folic acid when combined with the glycine produces a glycolic acid. Chenodeoxyfolic acid when combined with the glycine produces chenodeoxyfolic acid. So when the glycine is combined with the benzoic acid produces the picoic acid. These are the conjugation reactions of the glycine. See here it is. So bile acids is formed from the degradation of the cholesterol. How bile acids are formed? Bile acids are formed from the degradation of the cholesterol. So cholesterol, here it is, undergoes the degradation to produce the 7-hydroxy cholesterol. This reaction is catalyzed by the 7-hydroxylase. 7-hydroxylase. See here it is. This 7-hydroxy cholesterol is undergoes the several reactions, produces the folic acid and chenodeoxyfolic acid. These folic acid and chenodeoxyfolic acid non-conjugated bile acids. Okay, non-conjugated. Up to here it is, they are not conjugated with the any other component. Okay, folic acid and chenodeoxyfolic acid. But here it is this folic acid when combined with the glycine produce the glycogenic acid. So the chenodeoxyfolic acid when combined with the glycine produce the glycogenodeoxyfolic acid. These are the conjugated bile acids. Okay. So here it is the glycolic acid and glycogenodeoxyfolic acid conjugated bile acids. Before that here the folic acid and chenodeoxyfolic acid non-conjugated bile acids. So when these non-conjugated bile acids are combined with the glycine, so produces the, here it is called the conjugated bile acids. The conjugated bile acids are glycolic acid, tarfolic acid, and chenodeoxyfolic acid, glycogenodeoxyfolic acid. But now we are talking about the only glycine conjugated reactions. That's why here it is the folic acid is when combined with the glycine produces the glycolic acid. When chenodeoxyfolic acid combined with the glycine produces the glycogenodeoxyfolic acid. These are the primary conjugated bile acids. Okay. So how importance of the glycine involved in the conjugation reactions? What is the importance of this conjugation with the bile acids? What is the property? Why it is required for the conjugated with the glycine these bile acids? So this conjugation is bile acids, they are increasing their amphipathic property. When the bile acids are conjugated with the glycine, increasing their amphipathic property. So that's why it is a necessity for the conjugated with the glycine, these bile acids. Okay. Now see the another and last one is biosynthesis of the creatine. Biosynthesis of the creatine. Glycine is involved in the synthesis of the creatine. So where the creatine is present? So especially is present in the tissues. That is example muscle, brain, blood. So creatine is present in the muscle, brain, and blood. It is present in as a high energy compound and also free form. So creatine is available in as a high energy compound and also free form. So high energy compound example phosphocreatine. So phosphocreatine is a high energy compound. So it is stored in the muscle. Okay. So creatine is available in high energy compound and also free form. So that is nothing but free creatine. So but here it is also required 3 amino acids for the synthesis of the creatine. So glutathione also synthesis of the glutathione, they require the 3 amino acids. Even creatine also required the 3 amino acids. There, that means the synthesis of the glutathione is required the, here it is a cysteine, glycine and glutamate. Cysteine, glycine and glutamate. Okay. So there, here it is the synthesis of the creatine, glycine, arginine and methionine. So here it is common amino acids both in synthesis of the glutathione and synthesis of the creatine is a glycine. Glycine is a common amino acid that is synthesis of the glutathione and synthesis of the creatine. So there also is 3 amino acids required for the synthesis of the glutathione. So synthesis of the creatine also required for the 3 amino acids. But here it is glycine, arginine and methionine. There, here it is a 2 Gs, that is a glycine, glutamate and cysteine are required synthesis of the glutathione. Okay. Now, see the synthesis of the creatinine occurs in, so these reactions is occurs in my kidney, and here it is the liver. The first reaction, so glycine, here it is the originine. See here it is. This guanido group of originine transferred to the glycine produce the guanidoacetate. Guanidoacetate. So originine transfer rates guanido group of originine combined with the glycine transfer to glycine produce the guanidoacetate. This reaction is catalyzed the transaminase. This reaction is occurs in kidney. So the formation of the guanidoacetate is occurs in the kidney. That means glycine is combined. Guanido group of originine is combined with the glycine produces the guanidoacetate. This reaction is catalyzed by the transaminase. This reaction is occurs in kidney. Okay, this guanidoacetate, that means is S-adenosine methionine. S-adenosine methionine transfer its methyl groups with the guanidoacetate. S-adenosine methionine, so transfer its methyl groups to the guanidoacetate produce the creatine. So this reaction is occurs in the, here it is the liver. This reaction is catalyzed with the methyl transferase. So ultimately creatinine occurs, synthesis takes place in the liver. Okay. Now this creatine, here it is convert to the creatine phosphate. Undergoes the reversible phosphate produces the creatine phosphate. This reaction is catalyzed by the creatine kinase. This creatine phosphate as phosphocreatine is a high energy compound stored in muscles. Creatine phosphate is a, here it is a high energy compound stored in muscle. Okay. Now see the formation of the creatinine. So this creatinine undergoes the cyclization reactions. Now the creatinine undergoes the cyclization reaction, produce the creatinine. This creatinine is a excretory product. That means creatinine is a anhydrous form of creatine. So generally, so Viva is asking, what is the creatinine? Then you have to give the answer, anhydrous form of the creatine. Creatinine is a anhydrous form of creatine. Anhydrous means is removal of water molecule. So anhydrous means removal of molecule. So creatinine is a anhydrous form of creatine. So it is creatinine formed from the cyclization reactions of creatine or creatine phosphate produce the creatinine. This creatinine is a excretory product. Okay. So the synthesis of the glycine. So when the glycine originine guanido group of arginine transferred to the glycine, it will produce the guanidoacetate. This reaction is occurs in the kidney. So here it is the S-adenosine methionine transfer donates its methyl group to the guanidoacetate, produce the creatinine. This reaction is occurs in the liver. So ultimately synthesis of the creatine takes place in the liver. This creatine is converted to the creatinine phosphate. This is a high energy compound. This creatinine phosphate is stored in the muscle. That means creatinine is stored in the muscle in the form of is creatine phosphate. Creatine phosphate is a high energy compound. So high energy compound, for example, creatine phosphate is a high energy compound. See here it is that this creatinine is formed from the cyclization reactions of creatine or creatinine phosphate. So that means is creatinine is a anhydrous form of the creatine. Okay, so creatinine is a excretory product. Now see the clinical significance of the creatinine. So see the normal values of the serum creatinine and creatinine. Serum creatinine normal value is 0.2 to 0.6 milligrams per deciliter. Serum creatinine is 0.6 to 1 milligrams per deciliter. So urine creatinine is 0 to 50 milligrams per day. So and urine creatinine is 1 to 2 grams per day. See here it is difference between the, here it is units, serum creatinine and urine creatinine. Creatinine and creatinine also. Here it is milligrams per deciliter, but here it's milligrams per day. Urine creatinine and creatinine is units are milligrams per day. Serum creatinine's units are milligrams per deciliter. Serum creatinine and creatinine units are milligrams per deciliter. Urine creatinine and creatinine's units are milligrams per day. One is urine creatinine is milligrams per day, creatinine is grams per day. Okay. Now, so the clinical importance of the creatinine and creatinine. Generally, the daily excretion of creatinine is usually constant, but here it is when the output of creatinine is increased through the urine is nothing but it's called creatinuria. Creatinuria means is increase the output of creatinine in urine is called the creatinuria. This creatinuria observed in muscular dystrophy, diabetes mellitus, hyperthyroidism, and starvation conditions. Metabolic disorders of the glycine. So the main metabolic disorder of the glycine is glycinuria and primary hyperoxaluria. Hyperoxaluria. So glycinuria and primary hyperoxaluria. So in glycinuria is a rare disorder, but here it is the serum glycine concentration is normal, but when it is so increased high amount is excreted in urine, that is known as a glycinuria. The amount of very high amount of glycine is excreted in urine is known as glycinuria. So the normal value of glycine is 0.5 to 1 gram per day. And primary hyperoxaluria, it is also the involved error. So high urinary excretion of the oxalate, we can observe the oxalate levels are increased, excretion of the oxalate through the urine is increased due to the mainly deficiency of the enzyme glycine transaminase. Due to the deficiency of the glycine transaminase. So this is the glycine. So glycine is a non-essential amino acid. Is involved in the synthesis of the heme, involved in the synthesis of the bile acids, and involved in the synthesis of the glutathione. So don't forget the main sources of the synthesis of the glycine is serine, threonine, glyoxylate, carbon dioxide, ammonia, and one carbon unit. One carbon fragment is required for the formation of the glycine. So glycine involved in the reactions of formation of the purine, involved in the formation of the one carbon metabolism, involved in the synthesis of the heme, involved in the synthesis of the creatinine. So okay, don't forget involved in the formation of involved in the conjugation reaction. That means is conjugation reaction is conjugated with the bile acids. Conjugated with the